Assalamu alaikum, sir. I'm Dr. Mariam.
Yes, Dr. Mariam.
I have a question for you. What are selectins and what can you tell me their clinical importance?
Right. So Dr. Mariam is here online with me and she is asking a question. Number one, what are selectin molecules? And number two, what is the clinical importance of selectins? Why to learn selectins, right? Dr. Mariam, basically selectins are one of the group of cell adhesion molecules, right? Cell adhesion molecule have basically four families, which are basic families, primary families. Number one, selectins. Number two is integrins. Number three is immunoglobulin superfamily. And number four is cadherins, right? Now, today we will be talking in detail about the selectins. So, what are selectins? Selectins are basically the smallest family of cell adhesion molecules. They have only three members. Selectins have only three members, and those three members are, there are P selectins, there are P selectins, there are E selectins, and there are L selectins. Now, these are basically adhesion molecules which are expressed, right, on the cells of the cardiovascular system, right? For example, P selectins. P selectins are primarily expressed on endothelial cells, plus they can also be expressed on platelets. Because they were originally discovered first of all in the platelets, that's why they are called P selectins, right? P selectins are present within the endothelial cells. For example, this is an endothelial cell, this is its nucleus. Within the endothelial cell, there are special granules, and these granules, which are called viable pallid bodies. So within the endothelial cells, there are viable pallid bodies. These viable pallid bodies have many components, including P selectins in their membrane. A very important component within the viable pallid body is von Willebrand factor. But we will not go in detail of von Willebrand factor here. We will keep on concentrating on the selectins. So P selectins are number one, present in the viable pallid body, present within the endothelial cells, right? And number two, they are present in the platelets. They are present in the platelets alpha granules, right? Alpha granules of the platelets. They also have P selectins. And what about E selectins? E selectins are exclusively can be expressed only on endothelial cells. But there is a very big difference in P selectins and E selectins. P selectins are preformed and pre-synthesized and stored within the viable pallid bodies of endothelial cells or alpha granules of platelets. They are pre-synthesized. Only under certain circumstances, like inflammation, they are expressed on the membrane. Opposite to that, E selectins are not pre-synthesized. They are actually, when endothelial cells are activated, then they start expressing the genes of the E selectins, and then they start synthesizing the E selectins, right? So it means as compared to P selectins, E selectins are not pre-synthesized, not pre-stored. They are only synthesized when endothelial cells are activated, and activated endothelial cells, especially when they are activated by interleukin 1 or tumor necrotic factor or other cytokines, then endothelial cells are activated and they start expressing the E-selectins. And when we talk about the L-selectin, L-selectins are basically expressed on the white blood cells, right? Especially, we should not forget L-selectins are very, very important for lymphocyte trafficking, right? They are also expressed on the lymphocytes. So in a brief way, we can say P-selectins are P for platelets. They are present in platelets and they are present in endothelial cells and they are preformed. In platelet, they are in alpha granules, and in endothelial cells, they are present in Weibel palette body. These Weibel palette bodies also have a very important component, von Willebrand factor. E-selectins are only expressed on the activated endothelial cells, otherwise normally they are not expressed. And L-selectins are naturally expressed on the white blood cells, right? Now, what are exactly these selectins? Structurally, if we talk about, right, what are these selectins, right? These selectins are basically special type of glycoprotein molecules, and these glycoprotein molecules are passing through the membrane. Let's suppose this is a cell, right? Rather, I will make a smaller cell so that I'm going to draw the P-selectin. Let's suppose this is an endothelial cell, right? And if it is stimulated by histamine or thrombin, then Weibel palette bodies will move towards the cell membrane, and there they will express P-selectins. Now, what is the structure of P-selectin? Basically, P-selectin, number one, they have a very big extracellular domain, right? Overall, these cells are single pass. Single pass means as a glycoprotein, they pass one time through the cell membrane. So basically, they are transmembrane molecules, single pass transmembrane molecules, which are structurally glycoproteins. So let me draw their structure in detail. When we study from the extracellular component, first of all, this is their ligand binding component. When these cells are going to, this molecule is going to become adherent to the other molecule, right? Now this domain is, we call it lectin binding domain, and their binding depends on calcium. This is lectin binding domain. What is meant by the lectin? Basically, lectin binding domain means a domain of this molecule which loves to bind with carbohydrates. It means the other cell with which this is going to interact, that should express a receptor which has some carbohydrate in the receptor, right? Or we say this loves to bind with sweet carbohydrates. I will go into detail later, right? What kind of receptors they are. So lectin binding domain, which is actually carbohydrate binding domain, and this binding between this receptor domain and its counter receptor, that is the ligand, right? That binding depends on presence of calcium. Number two, just if you study it inward to the cell, then just under the ligand binding domain, there is another domain, and this domain is, yes, we call it epidermal growth factor like domain. Epidermal growth factor like domain. Then under it, there are some a short consensus sequence. And here I'm going to draw this is short consensus sequence, and in case of p-selectin, this consists of nine components, 9 units. But in case of e-selectin, this varies and it becomes shorter and it becomes 6 unit. And in case of L-selectin, this short consensus sequence. In case of L-selectin, it is only 2 units, right? It means when you move from p-selectin to e-selectin and then L-selectin, because consensus sequence progressively becomes smaller, so selectin molecules progressively become smaller. So if someone asks you the largest or longest selectin molecule, you will say p-selectin. If someone asks you shortest selectin molecule, you will say that is L-selectin, right? Now, next important component is, this one. This is transmembrane component. What we call it? Transmembrane. And this is the last component, which is called cytoplasmic tail. So how many unit? What is selectin? Again, I will repeat. Selectin molecules are adhesion molecules which are specially expressed in the cells of cardiovascular system, and they are classically can be expressed on the p-selectins can be expressed on platelets and endothelial cells. E-selectins can be are expressed on the endothelial cells only, and L-selectins primarily they are present, mainly they are present on the leukocytes, white blood cells, especially on lymphocyte. Basic structure, that is one pass transmembrane glycoprotein molecule, and its extracellular component, we can, if you study the outermost part, is lectin binding domain, which will bind with its receptor and make this other cell adherent to this cell. Then we have epidermal growth factor like receptors, then short consensus sequence, and then there is cytoplasmic tail, right? Now, after this basic introduction that what are selectins, let me go to this thing, that what is their importance, right? I will discuss in detail structure and especially functions of these molecules later, right? Here I'm just going to mention very briefly why these molecules are becoming very, very important in learning of modern medical sciences. Right, number one, selectin molecules, the first importance is they play a very important role in recruitment of leukocytes. How? During inflammation. You know, when inflammation is there in tissue, then endothelial cells are activated, right? First of all, endothelial cells can be activated by histamine and thrombin. When endothelial cells are activated by histamine and thrombin, then endothelial cell from the viable palate body express p-selectins, right? These p-selectins will appear very rapidly after the inflammation, maybe within few minutes, right? And then other chemical mediators are produced like cytokines at the site of inflammation. For example, tumor necrotic factor alpha or interleukin 1, they will act on also on the endothelial cells, and then endothelial cells will start synthesizing, e-selectins, right? They will start synthesizing e-selectins. So it means when inflammation occurs, p-selectin is expressed on the endothelial cell at very early time, within few minutes, under the influence of histamine and thrombin. But when there is when there is little more time to the inflammation and there is accumulation of cytokines, then cytokines activated endothelium, right, that will express E-selectins, right? And L-selectins, I already told you that they are naturally present on leukocyte. Now what happened? What we can say, during inflammation, due to expression of these selectins, endothelial cells become sticky. Sticky to what? Actually, these selectins, P selectins and E selectins, their counter receptors or ligands are present on white blood cells. For example, on neutrophils, right? I will discuss their counter receptors in detail later. Now, what happens? When inflammation occurs, endothelium become activated. Activated endothelium expresses many types of adhesion molecules. First of all, P-selectin, then E-selectins, and I will teach you later that later on they also express the ligands for integrins. Now, due to this process, endothelial cell becomes adherent. They become sticky. Because sticky to what? Sticky to white blood cells, especially neutrophils and then macrophages. Why? Because when endothelial cell express selectins, the selectins receptors are present on leukocyte and leukocyte start binding with the endothelial cells via the selectins, right? And selectins and its receptor bind very short, for very short time, and they bind with very low affinity. Because they bind loosely, due to that reason, when neutrophil bind with some selectins, right, that is binding transiently and weakly, when blood flow comes, it will roll over. Then it will bind with more selectins, right? But the other group of selectin is also what? Making weak bonds and transient bond. When blood flow will come, it will further roll. So selectins are playing a very important role in cellular events of acute inflammation. Rather, the very early event that is called leukocyte rolling. So what is leukocyte rolling? That when there is activated endothelium, and activated endothelium is expressing P-selectins and E-selectins, and their counter receptors are present on the white blood cells in inflamed tissue. So when white blood cells or leukocytes, more specifically, if it's early time of inflammation, neutrophils, right, then they are bound with the activated endothelium via the selectins. Binding is very, you have to remember, binding is very weak and binding is very transient. And this weak binding, for a short time, they are held, but what will happen? Blood flow will come and this pressure will roll it forward, right? So leukocyte will roll over the sticky endothelium. Leukocyte will roll over the sticky endothelium, or we can say leukocyte will roll on the selectin expressing endothelium. Selectin expressing endothelium, right? What is the purpose of this rolling? The purpose of this rolling is to make the leukocyte flow slow. Because leukocytes are moving through the inflamed tissue, we don't want from arterial side the leukocyte come into microcirculation and from venous side they escape. We want leukocytes to extravasate, to go out of vascular system. For this purpose, first step is, there are many steps of inflammation. One of the steps is making the endothelium sticky, and leukocytes are temporarily binding, attaching, detaching, rolling, attaching, detaching, rolling. So they attach, they detach and roll. This process helps neutrophils to slow down. When they slow down, right, they will eventually find, leukocyte will find interactions between integrins and their counter receptors on endothelial cells. Integrins are expressed on leukocyte, right, white blood cells, and their counter receptors are expressed on the endothelial cells. When integrin bind with the counter receptors, this is relatively strong adhesion, and rolling leukocyte are arrested there, right? But this again we will discuss in detail later. So this was the role of, this is how selectin lead to you can say sticking between the two cells. One cell will be expressing the selectin molecule, and other cell which is going to stick should be expressing another glycoprotein which is having oligosaccharides, because I told you selectins love the carbohydrates. Later on I will tell you that, for example, this is endothelial cell, this is neutrophil, right? Neutrophil, if this is showing selectin, neutrophil will also show a glycoprotein, but this glycoprotein will have a short chain of carbohydrate that is called oligosaccharide, that only has four monosaccharides. So also we call it tetrasaccharide. This tetrasaccharide, which is present on neutrophil glycoproteins, this has a very special type of two units. Number one, sialic acid, and number two, fucose. So we say that when there are two cells, if they are going to stick with each other via the selectin system, then one cell should be expressing the selectins and other cell should express the glycoprotein which are having some short saccharide chain. So we can say the other cell should be expressing oligopolysaccharide attached with glycoproteins, right? So what is this? This is neutrophil. It is expressing glycoprotein. Glycoprotein at its tip has a short oligosaccharide, right, or tetrasaccharide, and this tetrasaccharide has two special important units. One is fucose and other is what? Sialic acid. For example, let me show you. Here is endothelial cell, right, and here is your selectin molecule. Then leukocyte, for example, this is neutrophil, right, neutrophil. It should also express glycoprotein. Usually for p-selectin, they show two glycoproteins which are held together by disulfide bond. These two glycoproteins, right, are having a short, I should say, oligosaccharide, right, and that oligosaccharide is having two very important components. One is fucose, this F is for fucose, and other is, yes, sialic acid. This is also fucose and sialic acid. Now this molecule, in simple word, what we should call it? In a very simple word, we should call it that this is counter receptor for p-selectins, right? This whole structure is counter receptor for p-selectin, or we can call it is ligand of the p-selectin, or when we say biochemically, we say this is a glycoprotein expressed on the leukocyte. This is homodimer because these are two molecules which are similar to each other, identical to each other, but held together by disulfide bond. So these are glycoprotein homodimer, which is having at its extracellular domain, it is having oligosaccharide which express the fucose and sialic acid, right? So there are other names for it, and these are sialyl polysaccharide, or sialylated oligosaccharides. Sialylated oligosaccharides which are bound with the, what is this, glycoprotein, right? This sialylated oligosaccharide has another name, that is SLEX. That is sialylated Lewis X domain. What is this called? SLEX is this unit, right? That is what? Sialylated Lewis X domain. So we can say that here sialylated Lewis X domain is acting as sweetie. It is that sweet point where selectin molecules binding domain will stick, right? And this molecule is because for p-selectin it is given another name, p-selectin glycoprotein ligand 1, PSGL1. P-selectin, yes, what is it? Glycoprotein. It binds with the p-selectin, right? Glycoprotein ligand 1, right? So what I wanted to say, that whenever there is inflammation, endothelium is activated. Activated endothelium expresses the selectins. P-selectins are preformed and E-selectins are induced, and they appear on endothelium after 1 to 2 hours. The purpose of these selectins is that they try to slow down the movement of the white blood cells or neutrophils. How do they do it? Basically, selectins bind with the receptors on, for example, this p-selectin, they bind on the receptors on the neutrophils, and these counter receptors or ligand of the p-selectins are glycoproteins with special type of, you can say, carbohydrate unit, which should have SLEX. SLEX is what? Sialylated Lewis X epitope, right? So this was the role of selectins in inflammation, that it helps in recruiting the white blood cells. Then there is another role of selectin, that is L-selectins. As I told you, L-selectins are expressed on the white blood cells. Especially I would mention that L-selectins which are being expressed on the lymphocytes, right? What happens? These lymphocytes, right, when they are circulating in our circulatory system, right, when they reach to the peripheral lymphoid tissue, especially in the lymph node or in the MALT. MALT means mucosa associated lymphoid tissue. Again, for example, GIT associated, in GIT mucosa associated lymphoid tissue, or respiratory mucosa associated lymphoid tissue. So these are two examples of MALT. What happens? Lymphocyte expressing the L-selectins, when via the circulation they are carried to lymph nodes or to the Peyer's patches, for example, right? There they stick to the endothelium. For example, in lymph node, when lymphocyte reach to high endothelial venules, these are special venules which are present in lymph nodes, and they show plump or cuboidal endothelial cells. Remember, normally endothelial cells are flat, right? These cells express the receptors, rather counter receptors or ligands for the L-selectins. So when lymphocytes reach there, they bind with endothelium of high endothelial venules. They bind there, and then eventually they extravasate, they go into follicular area of the lymphocyte, and within the lymphocyte they interact with the different antigen presenting cells. So they can be activated if their relevant antigen is present, and they can be proliferating, right? So, and eventually those, for example, if antigen A is present in lymph node and such lymphocyte reaches that lymph node, right, selectins, L-selectins on lymphocyte and their counter receptor or ligand on the high endothelial venule, they will interact, stop that lymphocyte and help it to eventually, some other molecules are also involved, to extravasate. And out of the lymphoid vascular system, this lymphocyte, which is will shift to the follicles. And there, if there is antigen presenting cell is presenting A antigen, and if the lymphocyte have TCR, T cell receptor for A antigen, then they will interact with each other with the help of other molecules also. Eventually this specific lymphocyte, which has ability to interact with the A antigen, that will multiply over there, proliferate over there and become mature and get ready to fight with that antigen. And then this lymphocyte will re-enter into circulation and it will tour all your body and find where is the A antigen carrying pathogen present, right? So it means that L-selectins are playing a role in circulation of special trafficking of lymphocytes, that lymphocytes, naive lymphocytes, those lymphocytes which are probably not previously exposed to the antigens, they are visiting again and again lymph nodes or Peyer's patches, right? And they're looking for such antigen presenting cells which might present their relevant antigen. Then they have a chance to get stimulated and proliferated, and then they become millions of the cells, and eventually they re-enter into circulation looking for the antigen. So we can say the second function of the selectin is that it helps in lymphocyte trafficking within the body. It is not other trafficking you hear, right? This is lymphocyte trafficking. It is not any kind of crime going on. So up to now we learned 2 points about the importance of selectins. Selectins are adhesion molecules, right? Number one, during the, for example, in acute inflammation, they help to capture the white blood cells by making them adherent, initially as rolling and early adhesion, and that is followed by more strongly sticky cells like integrins and their counter receptors, and eventually white blood cells will extravasate. So number one function of the selectins is that it acts as early adhesion molecules during the recruitment of leukocyte, for example in acute inflammation in early stages neutrophils. Number two, I told you that these L-selectins, they are present on almost all white cells, but I'm specially mentioning they are present on lymphocyte, right? And they help in trafficking of lymphocyte, that how lymphocytes, when I just explained how lymphocytes expressing the L-selectin, when they are passing through lymph node or Peyer's patches or other mucosa associated lymphoid tissue, these L-selectin expressing lymphocytes will bind with that lymphoid microcirculatory endothelial cells. There are special endothelial cells which are waiting for lymphocytes in the microcirculation of lymphoid tissue. What are those special cells? Those special cells are high endothelial cells. So these venules are called high endothelial venules. From there, these lymphocytes will go extravasate and try to interact with different antigen presenting cells so that they can see that there is their relevant antigen or not, right? These are two functions I told you, that recruitment of leukocyte and trafficking of lymphocyte. Then I can tell you some extremely important, clinically relevant situations in which now we have discovered in modern molecular science, we have discovered the role of selectins. For example, in post-ischemic reperfusion injury. Now, what is post-ischemic reperfusion injury? For example, if coronary artery has a thrombus and that result in ischemia to a part of myocardium. That ischemic myocardium is suppose severely injured, right? And that becomes suppose very severely ischemic. If that thrombus is spontaneously dissolved or by our drugs, and again that vessel open suddenly, we say there is reperfusion. Even though remember, there is benefit of reperfusion in early time that it rescues the ischemic tissue and prevents the enlargement of the infarcted tissue. But there is evidence that reperfusion can do paradoxically damage to the ischemic tissue. Why I say paradoxically? Because when you are doing reperfusion, blood flow again start to ischemic tissue. Primary logic tell us that tissue should be happy because it is receiving the blood and oxygen. But there is a special paradoxical phenomenon seen that is called post-ischemic reperfusion injury, in which what happens when blood flow again start in ischemic area, whatever ischemic area, it is myocardial tissue or it is renal tissue or wherever. When reperfusion occur, lot of oxygen arrives and unfortunately metabolism of ischemic tissue is altered and that molecular oxygen is converted into oxygen derived free radicals, superoxides that damage that tissue, number one. Number two, reperfusion might produce microcirculation vasoconstriction that may do damage. But during our discussion right now, I want to say that when there is zone of ischemic tissue, the microcirculation there, endothelium is activated. And endothelium start expressing what? Selectins. So, in severely ischemic tissue, if endothelium is still alive, it becomes activated due to maybe histamine or maybe due to other cytokines, and these activated endothelial cells in ischemic area, right, they start expressing selectins. So what does it mean? It means when reperfusion will occur, neutrophil will stick there. And if too much neutrophil is binding there, slowing down, rolling and slowing down, and then under selectins, they will do rolling and slow down of their movement, right? Eventually, they will bind with integrins and then they will go extravasate through the help of special molecules called PECAM or CD99, right? Or so they extravasate these neutrophils. Is it good or bad? This is a very important question. What happens in ischemic tissue, again, is in ischemic tissue what is happening? That injured endothelium is activated and it expresses what? Selectins. And if the reperfusion occur, the new arrival of neutrophil love to bind there, selectin will do rolling and slowing down, and eventually these neutrophils will bind there and they will extravasate, and these neutrophils become activated and they produce destructive products. You know, neutrophils can produce reactive oxygen species. Neutrophil can produce by degranulation a lot of lysosomal components and proteolytic enzymes, and also they produce many pro-inflammatory chemical mediators activated. So what happened? All these things damage the tissue which was already ischemic. This type of injury is called post-ischemic reperfusion injury, and you have to remember that selectins also play a role in that by capturing the neutrophils and enhancing the neutrophil mediated injury in ischemic area where reperfusion occur, right? Then there are more very sad situation, chronic inflammatory disease. In chronic inflammatory disease, right, I'm not going to teach you the pathogenesis of all chronic inflammatory diseases. Here I just going to tell you about selectins. In chronic inflammatory tissue, for example, in patients where there are chronic inflammatory lesions, for example, there is atherosclerosis or rheumatoid arthritis or inflammatory bowel disease, especially Crohn's disease, or SLE, systemic lupus erythematosus, or other chronic inflammatory diseases. Whenever I say the word chronic inflammatory disease, it means inflammation going on for a long time. What does it mean? It means in that tissue cytokines are being produced. It automatically means that these cytokines which are present being formed in chronic inflammatory focus, these cytokines will also activate and injure the local endothelial cells, and endothelial cells will express selectins, and selectins will again stop white blood cells. So what happens in chronic inflammatory diseases? Injured and activated endothelium, which is activated due to chronic presence of the cytokines, expresses lot of selectins, and these selectins bind with the leukocyte, and these leukocytes further enhance the injury, right? Why I'm saying these things? Because now lot of research is going on to develop such drugs which can either block the selectins or their receptors. Even some drugs are in preclinical situation, right? Then last but not the least, very dangerous role of selectins. You must be knowing cancer cells, malignancies. When there is primary malignancy, there are many molecular mechanisms which help the malignant cells, or now onward I say cancer cell, where cancer cells detach from the primary mass, and these cancer cells enter into local microcirculation. Then they are carried through the circulation to distant area, or through the lymphatic to the distant area, or through the serous membrane to the distant areas. And in distant area, these cancer cells may bind and start a new colony of malignant cells, which are called secondaries or metastasis. Now, there are many molecular mechanisms which orchestrate all this role, that detachment of the malignant cell from the primary mass and survival in the circulation, and eventually extravasate into some other tissue at some distant place and proliferate there. But I would love to hear mention, selectins are also here working against us. These cancer cells are very clever, rather I should say they are very cunning. Cancer cells express lot of sweeties on their membranes. Cancer cells express lot of mucin-like molecule. They express lot of carbohydrates on their surface. Actually, among those carbohydrates, cancer cells express lot of counter receptors or ligands for selectins. So what happens? When from the primary mass, if a cancer cell separate, as soon as it come into circulation, activated platelets will express the p-selectins, and p-selectins will interact with the cancer glycoproteins with oligosaccharide, and this cancer cell will bind lot of, what is this? Platelets. Cancer cells will bind lot of platelets, and these platelets, cover of the platelets over the cancer cell is protecting the cancer cell and circulatory system from other immune cells which might damage or kill the cancer cell, and they help the cancer cells to survive in circulatory system. Eventually what happens, that these cancer cells reach to a tissue, distant tissue, where they find favorable environment. Let's suppose in distant tissue they find another type of selectins. Now, when cancer cells expressed, they were sticking around them platelets via p-selectins on the platelets and sugars present on the cancer cells. And cancer cell moved through the circulatory system safely. And when they reach to point where they want to extravasate and produce the metastasis, first they need to slow down in that circulation of that target tissue, target metastatic tissue. To slowing down, they will bind. What will happen? Cancer cell will reach to that tissue. Cancer cells will damage the endothelium. Endothelium will express p-selectin molecules, and also cancer cells will activate by producing cytokines, might activate target metastatic site endothelial cells which express E-selectins. So this P and E-selectins on the target site will help the cancer cell to roll and eventually stick and bind at the target tissue, where they will eventually, what they will do? They will extravasate and produce secondaries. Here I will give you a very classical example. You know, in prostatic cancer, the cells love to go into bones, right? That malignant cell produce secondaries in bones along with other tissues. They have seen that actually endothelial cells in microcirculation of bone express a lot of selectins, and cancer cells are expressing those sweeties, counter receptors or ligands for those selectins, and these selectins help the cancer cell to settle in the bone, to become adherent with endothelial cells of the microcirculation of bone marrow or bone, and make metastasis there. So it means that now we can generate some drugs. Actually work is going on already, that we are trying to make some anti-cancer drugs which can help to reduce the chances of metastasis by binding the selectins or their receptors, right, on the cancer cells. So these were 5 points I told you about clinical importance of selectins, right? 5 points, right? Number 1, selectins play a role in recruitment of white blood cells during inflammation. Number 2, selectins, especially L-selectins on lymphocyte, help in the trafficking of lymphocyte. Number 3, selectins are expressed in ischemic microcirculation, ischemic tissue microcirculation, and they play a role in post-ischemic reperfusion injury. Number 4, selectins are expressed in lesions of chronic inflammatory diseases, where these selectins bind more neutrophils and more monocytes and more other inflammatory cells or white blood cells, and further perpetuate the chronicity of the disease, right? And 5th was that selectin mediated binding between the malignant cell and the platelets help the malignant cell to survive during the circulation, and also when cancer cell reach to their target metastatic tissue, there they will bind with microcirculation via selectins, and once they bind over there, they can lead to extravasation and metastasis development, right? Now we will take a break. After the break, we will discuss in detail p-selectins and their ligands. Then we will compare p-selectin versus E-selectin versus L-selectins, right? We will discuss in detail, and receptors of selectins in the next lecture. Right? Thank you very much.